So this is the fifth lecture of Bio 103, and this lecture has to do with the important macromolecules of the cell. This slide here gives a summary table of the biological macromolecules that we are going to focus on today. I would like you to keep in mind the structure and function of these macromolecules, specifically what kind of bonds are formed in each of these macromolecules, how they're formed, and how they're broken. You should be able to state what monomer or building blocks are used to create these macromolecules, and you should be able to give examples and know their role in the cell. So we have four main biological macromolecules: carbohydrates, proteins, nucleic acids, and lipids. Your carbohydrates are going to be based off of your simple sugars. They have many different functions in the cell: energy storage, they provide cell surface markers, etc. Your proteins are very important in the cell. They're based off of your amino acid groups, and they're going to be important in catalyzing a wide variety of reactions in the cell, also providing some structure, and they are also very important in cell signaling. Your nucleic acids are important for storing genetic information, as in the case of DNA, and RNA is very important in expressing those genes. You have a variety of nucleotides that are present within the cell that help make up these nucleic acids. And finally, lipids, our last group of macromolecules, they're going to be built off of fatty acid chains as well as glycerol. They are very important in providing energy for the cell and also providing the base structure for cell membrane. They can also be involved in cell signaling, as steroids are very important in hormone production. So we'll start off with our first macromolecule group, the carbohydrates. Carbohydrates are built off of your simple sugars. A simple sugar is called a monosaccharide, or mono meaning one sugar. Some common examples of simple sugars are glucose, galactose, ribose, and fructose. You can refer to their structures in the diagram to the right. A triose is a sugar that is comprised of 3 carbons, pentose has 5 carbons, and a hexose sugar has 6 carbons. So by knowing the base structure of a sugar, you can determine how many carbons are involved in creating that sugar. Sugars can also be classified as ketoses or aldoses. And if you remember your functional groups, this would indicate the position of those functional groups. A ketone would have the C double bond O group or carbonyl group in the middle of the structure, whereas an aldose would have it at the end. And you can see in the diagram they have various different types of aldoses and keto sugars for you to refer to. Sugars, if you remember the versatility of carbon, sugars can be present in a linear form, but they can also be present in a ring form. Most of the time in aqueous solution, sugars will exist in the ring form, such as glucose here. When you have glucose in the ring form, it can exist in 2 different isomers, the alpha and the beta form. If you take a look at the alpha glucose, you can notice that there's different carbons that can be numbered on the sugar molecule. If you refer to the 1 in the diagram, that refers to the first carbon in the chain. That carbon has a hydroxyl functional group. In the alpha position, it is found below the 1 carbon. If you take a look at the beta glucose to the right, you will see that that hydroxyl group ends up being shifted upwards of that number 1 carbon. So that's a base structural difference between the alpha and beta glucose. Remember that even though this is a small structural change, they can have different properties within the cell. Disaccharides are 2 sugars that are joined together by what we call a glycosidic linkage. So considering the sugars as your biological macromolecule, the important linkage in that structure would be the glycosidic linkage. This linkage is produced by a dehydration reaction. Dehydration reaction means that a water molecule is going to be removed from the 2 components to then create that glycosidic linkage. Some important examples of disaccharides would be maltose. Maltose is formed when you join 2 glucose molecules together. Sucrose consists of glucose and fructose, and lactose consists of glucose and galactose. If you take a look at the diagram here, it's important to note that this just depicts the formation of the glycosidic linkage. So if you see between two glucose molecules here, a water molecule is going to be removed and the glucose molecules are going to be joined together to form that glycosidic linkage. You will notice that we refer to that glycosidic linkage as a 1,4 linkage. This means that the carbon number 1 in the first glucose molecule is going to be joined by an oxygen to carbon number 4 on the adjacent molecule. It's important to keep in mind that when we have a glycosidic linkage, we can glean a lot about that linkage by looking at what numbers are involved in describing that linkage. So a 1,4 linkage refers to carbon 1 and carbon 4. Similarly, a 1,2 glycosidic linkage would mean that the carbon number 1 of glucose is then joined to the carbon number 2 on the adjacent fructose molecule in the creation of sucrose. Similarly, if we wanted to reverse this reaction and disrupt the glycosidic linkage, we would do the opposite, meaning that we would take a water molecule, apply it to the glycosidic linkage, usually using an enzyme, and that would serve to break apart that glycosidic linkage and return those two sugars to the monomer state. Here is another dehydration reaction. If we're taking a look at the formation of sucrose, once again, sucrose is the formation of a glucose and fructose monomer being put together in a glycosidic linkage. So we have glucose on the left added with fructose. Once again, we have the removal of water or a dehydration reaction, and that then leads to the formation of the 1,2 glycosidic linkage. Keep in mind that the 1 refers to the 1 carbon, and the 2 refers to the 2 carbon on fructose. There is a way in numbering these carbon rings. I am not asking you to be responsible for knowing how to number the carbons. That is if you take a specific chemistry course, but for the sake of this course, you should be familiar with what 1,4, 1,2, 1,6. If we're referring to a glycosidic linkage, you should know that that refers to the carbons that are involved in creating that bond. There are many types of polysaccharides that are important in the cell. Polysaccharides are long polymers of different types of sugars. These are very important for energy storage in the cell. Good examples of that are starch and glycogen. They have many different monomer glucose units that can be broken apart to then provide energy for the cell. They can also be structural, such as cellulose and chitin. Those are going to be important in maintaining cell structure. In this slide, we will take a look at the differences in these structures. Starch is considered to be the plant storage form of glucose. It is a polymer unit, which means that there's going to be many glucose molecules joined together by glycosidic linkages to form a starch molecule. There are two main forms of starch in plant cells, amylose and amylopectin. Amylose is considered to be the straight chain form of glucose, whereas amylopectin would be highly branched. Both are considered to be easily digested by all animals. Glycogen is considered to be the animal storage form of glucose. So in animal cells, we decide to store excess glucose in the form of glycogen. This is not possible in plant cells. Glycogen is also a polymer of glucose, specifically alpha glucose, but it's more branched than amylopectin. So if you were to consider how much energy is provided in these molecules, amylose would be the least amount of energy, while amylopectin would contain a medium amount of energy, and glycogen would definitely contain the most energy available to the cell. Cellulose is a structural polymer, so cellulose is very important in the formation of the cell wall. In the form of cellulose, cellulose is based off of beta-glucose as the monomer unit. As a result, this will not be branched, and it also will not form a helical structure. It will form fibrous-like structures to provide support. Cellulose, because of its beta-glucose linkages, is not considered to be digestible by animals. There are some animals that can digest cellulose. However, this is usually due to specific bacteria in their gut that can accommodate the digestion of cellulose. So they themselves cannot digest it on their own. They need symbiotic bacteria in order to be able to digest this material. Chitin is a structure that is important for the insect exoskeleton and also is an important component in the cell wall of various types of fungi. This is also based off of beta-glucose, but this is a polymer that consists of a nitrogen-containing form of glucose. This chitin structure is digestible by most animals, and this is because we do have an enzyme called chitinase that could help us digest this material. This image here shows the structural differences between these polymers. Starch can be broken down into amylose, the unbranched form, and the branched form amylopectin. You can see that there's a straight chain linkage, and this is because it is based off of alpha glucose as the monomer unit. Glycogen, remember, is the animal storage form of glucose, also based off of alpha glucose, and it can also be highly branched. Cellulose, on the other hand, is based off of beta glucose, and as a result, there is a change in the linkage. If you take a look at the figure at the bottom, you can see that the oxygen unit and with the position of that oxygen differs from the first to the second glucose molecule. So this means that the glucose molecules, when forming these bonds, actually goes from up to down to up to down. And this creates that change in that linkage that we are not able to digest. Because of this beta linkage, you also have these cellulose forming straight chain fibers. This gives a lot of structural resistance to the cell and provides a cell with a lot of support. This slide here illustrates the structural differences between the alpha and beta glucose. If you remember, carbon can exist as a straight chain molecule, and it also can exist in rings. So in the first image that we have here, we've got glucose in the straight chain form. When it forms a ring, it can form one of two isomers, either the alpha glucose form, where the hydroxyl group on the one carbon is in the bottom position, or the beta form, where it is located on top of the number one carbon. When you have beta glucose forming chitin, remember that that has a nitrogenous group added here, and you can see that in the bottom left figure. When you have these alpha or beta linkages forming into polymers, you can see that when you have the polymer based on alpha glucose, the linkage is pretty consistent throughout. That's the 1,4 alpha linkage in starch. And this is very easy to digest. Glucose monomer units will simply be broken off from the chain and will undergo metabolism. However, in the cellulose linkage, you can see that this is based off of the beta glucose, and the linkage actually goes from the top to the bottom position. And you can see the hydroxyl group will go from the top and bottom position, and the glucose molecule will actually be inverted when it's in creating this type of linkage. This is not digestible by the cell. Us animal cells, like I said before, we cannot digest this material. We simply do not have the enzymes available to us to digest it. So cellulose is considered to be roughage in your diet. You can only mechanically break it down into smaller pieces and smaller chains, but you cannot extract the energy from it. There are some organisms that do, because they consist of bacteria that can recognize this linkage, break it down, and extract the beta glucose units for energy. Now I'd like to introduce you to the second group of macromolecules that we will be discussing in this lecture, lipids. Lipids are considered to be your fats. Fats have little or no solubility in water. They are considered to be hydrophobic. Technically speaking, they're not considered to be true polymers, but they are still macromolecules made up of smaller monomer units. There are three main types of lipids that we will discuss. Fats, these are also called your triglycerides. Phospholipids, these are your diglycerides, and these are very important in forming the cell membrane. And finally, steroids. First, we will discuss the fats or triglycerides. Fats are comprised of a glycerol chain, and you can see in the diagram here, the glycerol chain refers to the gray area with the 3 carbons. Glycerol as a base structure has 3 carbons, and where you see that first O position, if you were just dealing with the glycerol chain, that would be an OH group. So a hydroxyl group is attached to each carbon. A triglyceride is named a triglyceride because it has 3 fatty acid tails. These fatty acid tails consist of long chain hydrocarbons. You can see in the image below that there's many carbon and hydrogen bonds that are present in each tail. These long chain hydrocarbons are carboxylic acids. That means that they will have a carboxylic group at the end of them. These provide a lot of energy that the cell can break down to fuel other processes. Fats are considered to be saturated or unsaturated. What that means is that when you have a saturated fat, that means all the carbons in that fatty acid tail are considered to be saturated with hydrogens. There's no carbon-carbon double bonds in that fatty acid tail. As a result, these compounds have higher energy value. They also have a higher melting point. A good example of this would be your butter or your lard. A good way to remember if you're dealing with a saturated or unsaturated fatty acid, when you have a saturated fatty acid, you have no double bonds in the tail. That means that all 3 tails can stack up very nicely on top of each other. Because they can get so compact, at room temperature, they are going to stop moving and they are going to form a solid at room temperature. So your saturated fats are going to be solid at this temperature. An important concept to understand is that if you take a look at your margarine, for instance, if you take a look at the ingredient list on a margarine container, you would notice that a lot of the oils are going to be hydrogenated. So you can have hydrogenated fatty acids. This is part of the manufacturing process. So you would have hydrogenated vegetable oil. That means that you are taking an unsaturated fat, you are loading it up with hydrogen, so you're exposing it usually to hydrogen gas. You are breaking those double bonds and making it into a saturated fat. This is mechanically done, this is man-made, and this is not naturally occurring in the environment. An unsaturated fatty acid is present when you have double bonds somewhere in the fatty acid chain. When you have these double bonds, they are usually present in the cis form, which means that the fatty acid tails are going to be on the same side and the hydrogens are also going to be on the similar side. This creates a kink in the tail. At room temperature, when you have a kink in the tail, that means that these fatty acids are not going to be able to stack up nicely with each other. There's going to be room for movement. As a result, these types of fatty acids are going to be liquid at room temperature. The lower energy that's available, these are much lower energy, and they also have a much lower melting point. Good examples of these types of oils are your olive oil, vegetable oil, etc. This slide here depicts the structural difference between saturated and unsaturated fatty acids. On the left, we have butter, which is a typical saturated fat. You can see the glycerol head region connected to the 3 fatty acid tails. Because there are no double bonds, all of these fatty acid tails can stack up nicely with each other, which means that they are going to be solid at room temperature. If you take a look at the unsaturated fat on the right, you can see that there's a presence of a double bond in the 3rd fatty acid tail. This creates a kink, which means that when multiple fatty acid tails and multiple triglycerides are stacked next to each other, they're not going to be liquid, they're not going to be solid at room temperature. Rather, this double bond is going to provide for movement, and that's going to mean that it is going to remain liquid at room temperature. Another important lipid that is present in cells is the phospholipid. This is a very important component of the cell membrane. The overall structure of a phospholipid has, once again, a glycerol backbone. But in this instance, that third carbon of the glycerol backbone is going to be bound to a phosphate group instead. This means that there are only two fatty acid tails. An important part to note is that that fatty acid tail, one of them will also have a double bond. What is important about the structure of a phospholipid is that the addition of this phosphate group creates a very polarized head region. It will also have a nonpolar region, which refers to the fatty acid tails. Remember, the fatty acid tails is made up of carbon and hydrogen bonds, which are considered to be nonpolar. When you place these phospholipids in water, because of these properties, they tend to want to form what's called a lipid bilayer. This means that these phospholipids will join together. The hydrophobic head regions will interact with each other, while the hydrophobic tail regions will want to interact with each other. This image here depicts what that phospholipid bilayer looks like. Note that the hydrophilic heads in gray are oriented towards each other, while the fatty acid tails in orange or yellow are also lined up. You can see that it is a bilayer because there are two layers of phospholipids. Finally, we get to the last group of important lipids, the steroids. Steroids are lipids that share a common carbon skeleton made up of 4 fused rings. Cholesterol is a very important steroid in animal cells. It is an important component for the animal cell membrane, and it is also a precursor to many other steroid hormones, such as estrogen, progesterone, testosterone, and cortisol. You can see in this figure here, this represents the small structural changes that would be present to convert cholesterol into a hormone such as testosterone. And once again, testosterone will have very different effects in the cell. Small structural changes can take that cholesterol and also make it into an estrogen hormone. Once again, that would have very different effects in the cell and in organisms. What is also important to note is that different people can have different responses to different hormones. Cortisol is an important stress hormone. So when people are stressed, some people process cortisol by losing weight, while as other people may process the cortisol and it may increase consumption of foods, which would lead to the gaining of weight. So even though you have the same hormone, you can still have differential responses between organisms.